Hi everybody, welcome back to our Chemistry 123 online lecture series. In the last class we looked at glycolysis, which is where we take glucose and turn it into two pyruvate molecules. In this lecture we'll take pyruvate then and look at its fate. Most of the time in humans it turns into acetyl coenzyme A and then feeds into the citric acid cycle. We'll also take a look at how glucose molecules are synthesized from other biomolecules, and we'll take a look at how glucose is stored as glycogen. Alright, so remembering where we are, glucose plus NAD ultimately makes two pyruvates. This is the reaction in glycolysis, the overall reaction. So we get two ATPs out of that, and we get two NADHs out of that. So if we looked at glycolysis, now how are we going to get that pyruvate into the rest of our citric acid cycle and electron transport chain? Alright, so pyruvate, under normal aerobic conditions in humans, animals, plants, and many microbes, pyruvate will ultimately again turn into acetyl coenzyme A, feed into the citric acid cycle like normal. Under anaerobic conditions in humans, animals, plants, some microbes, that pyruvate will turn into lactate during a process called fermentation. This is much less efficient, generates a lot less energy than turning pyruvate into acetyl coenzyme A, but sometimes that might be necessary. Some microbes use this metabolism obligately, meaning they can't do anything else. Finally, under anaerobic conditions, only some microbes, humans don't do this. Pyruvate will be turned into ethanol, again in a fermentation process. Alright, so what happens to pyruvate? Ultimately, a lot of it gets converted to acetyl coenzyme A. This occurs in the mitochondria. Ultimately, this consumes an NAD plus in its oxidized form, and this is supplied then by the electron transport chain further down. So this NAD molecule is again being recycled continuously. As the electrons get dumped off of it into the electron transport chain, that NAD in its oxidized form, NAD plus, heads back to pyruvate and gets then re-reduced. A carbon atom is removed in this process, so we generate CO2, and ultimately the product is coenzyme A from the free coenzyme A with the sulfur and hydrogen group on it. Alright, so again we can also convert to lactate. This occurs under anaerobic conditions. And finally we can convert to ethanol also under anaerobic conditions. So here's a summary of what we get out of this. Again, the middle there, lactate, and the right side, acetyl coenzyme A, are done by most eukaryotes, so animals, plants, humans, etc. Some, again, microbes obligately use the middle and left side of this diagram. So right away we can see some differences here. Pyruvate turning into acetyl coenzyme A generates an NAD, so we now have electrons to shuttle around. Pyruvate is a 3-carbon compound, and so if we're going to turn it into the acetyl group on acetyl coenzyme A, we need one less carbon, so that carbon now is oxidized and leaves as CO2. Once then we have acetyl coenzyme A that enters the citric acid cycle and proceeds how we studied last week. Alright, a little bit of a different form though, when we turn pyruvate into lactate or into ethanol, ultimately we consume that electron-rich NADH molecule, so it's not already going to be a little bit less useful energetically. That pyruvate, if it's going to turn into ethanol, again we also then have to remove a CO2. Acetaldehyde is already a 2-carbon compound, pyruvate's a C3, so off away that carbon goes. Alright, so let's look in a little bit more detail at pyruvate turning into acetyl coenzyme A. Alright, so ultimately this happens when the pyruvate from glycolysis in the cytosol crosses two mitochondrial membranes and ultimately arrives in the mitochondrial matrix. So we're ready to actually do work at the energy generating component of our cells. Alright, so When the mitochondrial matrix pyruvate is oxidized to acetyl coenzyme A, we have a four-step pathway, which we're not going to look at in any detail. It involves both oxidation and decarboxylation of pyruvate. All we really want to know is the overall reaction, what's going on. So here we have pyruvate. Remember, pyruvate is in its deprotonated form. This carboxylic acid here is ionized. It's got that minus charge because we're at physiological conditions. Remember, if we had this proton here, this would be pyruvic acid, but because of the local pH conditions, it's going to be deprotonated. In any case, we are ultimately shuttling electrons then over to this NADH and attaching the coenzyme molecule and generating a CO2. Alright, so we can now consider what happens. We've now fully connected our metabolism going from glucose all the way through oxidative phosphorylation. So to go from glucose then to acetyl coenzyme A, here is our reaction. So this is now considering the conversion of pyrite to acetyl coenzyme A all the way from glucose. And again, it's now then these acetyl coenzyme A's that will continue on into the citric acid cycle. Alright, so we do generate, again, some ATPs along the way in this step. We don't generate ATPs, notice, going from pyruvate just to acetyl coenzyme A. So these ATPs are from earlier in glycolysis. Alright, so let's look actually at how much energy we get in the form of ATP from the oxidation of glucose. So here we have glycolysis. We get a total of 2. Add that up. The oxidation of pyruvate, the next step, we don't get any ATPs. We get 2 ATPs from the citric acid cycle and a whopping 26 from the electron transport chain and oxidative phosphorylation. So right away we can see a couple of things. One, you'll often see that the amount of ATP generated per glucose molecule is 28, and in fact we have a figure in this later on where that's the case, and that's because this is actually a GTP. The difference is the base that's actually contained in it. It's guanine instead of adenine. But that's okay. It functions basically the same. It's still a high energy molecule. It still has those 3 phosphate groups. And so it functions much the same way as an ATP. So in this case, the text is counting it. So note here, if we only generate 2 ATPs though in glycolysis and none in the oxidation of pyruvate, well, something is going to happen where it looks like the citric acid cycle and electron transport chain are going to be a lot more energy yielding than fermentation processes, and it turns out that actually is the case. It's also worth noting now, this is the complete reaction pathway from going from glucose all the way then to CO2. So this is the complete reaction contained here of glucose plus oxygen makes CO2 plus H2O. That complete reaction yields 30 ATPs. Now let's have a look at the fermentation process. So in the absence of oxygen, like again may occur under vigorous exercise conditions, or you might again do this because you're a microbe living in an anaerobic condition, ultimately what's going to happen is you're going to have a buildup of NADH from glycolysis, which is going to negatively affect the rate of glycolysis, remembering that NADH is a feedback. So how are we going to then continue supplying the cells with energy during intense exercise? Well, ultimately what we have to do is generate more NAD+, in order to continue that process. So fermentation is the solution to that issue. So fermentation is a biological or biochemical and biological process by which NADH is oxidized to NAD+ without the need for oxygen. So in this case, we're taking the electrons from NADH, we're using that to dump them onto our pyruvate. When pyruvate becomes reduced then to lactate, that generates now NAD+ in the oxidized form, which is able then to be recycled back into the start of glycolysis, so it goes backwards, and then it starts the process over again. So specifically in lactate fermentation, we have an enzymatic anaerobic reduction of pyruvate to lactate. So remembering NADH is electron or hydrogen rich, well, it's both. And so remembering a reduction is the addition of hydrogen molecules. There are, again, other definitions, but that's the one most relevant to this. So we're taking this hydrogen and we're sticking it on pyruvate, and when we do that, it becomes reduced then to lactate. All right. So again, why we're doing this is to convert this molecule. And so when we convert that, we're able then to continue with glycolysis. That's the whole point of anaerobic fermentation. Also, as we'll see in a minute, we can convert lactate back to pyruvate when aerobic conditions return. So when oxygen's back around, we'll recycle that lactate in order to continue the process. Ethanol fermentation, again, works totally analogously. We're also, again, converting NADH to NAD+. Ultimately, then this pyruvate, instead of being reduced straight away, it's decarboxylated to acetaldehyde and then reduced to ethanol. So this is a step where we generate CO2. So again, this is not used by humans because we wouldn't be able to recycle that lactate back to pyruvate, but it's, again, a strategy for microbes to use. All right, so let's look at aerobic versus anaerobic carbohydrate metabolism and how that affects energy. So during, again, aerobic conditions, while we're breathing air, not exercising vigorously, we convert then the conversion of glucose to 2 pyruvates produces 2 ATPs and 2 NADH that's produced in the cytosol, then leads to the production of 3 ATP. Once we then take that acetyl coenzyme A, shuttle it through the citric acid cycle, the rest, and oxidative phosphorylation, we generate then a total of 28 ATP, 2 GTPs for a total of 30. Right? Okay, so we get 30, we'll just call it 30 ATPs out of 1 glucose molecule under aerobic conditions. All right, so when we're, again, running around really quickly, there's not enough oxygen around. The conversion of glucose to 2 pyruvates produces 2 ATPs. Okay, so what are we going to do from there? Well, we can't do anything with it. We can't generate any additional ATP from lactate, and we don't use ethanol, because neither of them continues on to the common metabolic pathway. All right, so if we look here, 28, or again 30 from that GTP, versus 2, that's not really very good. So what that actually means then is aerobic oxidation of glucose is 15 times more efficient per glucose molecule than anaerobic processes. So again, there are more, and this is a sort of interesting aside, there are more total cells on Earth to which oxygen is a deadly toxin than there are cells which accept oxygen. Well, why is that? Since it's such an obviously better energy source, look how much ATP we're generating. Well, that has to do with Earth history. There wasn't oxygen around in any appreciable abundance in the atmosphere until about 550 million years ago. So if there's no oxygen around, well, there's no oxygen to drive aerobic metabolism. It's that, again, oxygen is a very, very high energy yielding compound when coupled to carbon molecules. So again, glucose plus O2 is a very high energy yielding reaction. Glucose to lactate or glucose to ethanol is not a very high energy yielding reaction. And so we see that manifest in then ATP. And so why would you bother doing anaerobic metabolism at all? Well, it's, again, it has to do with the evolution of organisms on Earth, the fact that we didn't always have oxygen around. All right, so we can now go from glucose all the way to CO2 under normal conditions. We can go from glucose to lactate. We're going to spend not any more time looking at glucose to ethanol. So what else are we going to then consider? Well, the Cori cycle is a biochemical process in which ultimately pyruvate is converted to lactate, lactate is converted back to glucose, so that we're going to recycle lactate that's generated during anaerobic metabolism. All right. So ultimately, then, lactate is formed in muscle cells during strenuous exercise under anaerobic conditions. That lactate will then diffuse out of cells passively. It's a small enough molecule out into the bloodstream, and it's transported to the liver. In the liver, the lactate is then converted to glucose, and so we're then able to restart the process. All right, so that lactate gets converted to pyruvate, and then pyruvate then gets converted to glucose by the process gluconeogenesis, which we're going to look at in just a minute. All right, so that glucose can then be shuttled back to the muscle tissue to be converted to pyruvate, and so we can continue generating ATP aerobically. So in this way, we're again recycling lactate that we've generated in order to gain all of the energy that we can out of it. It does take some energy to convert lactate to glucose, so it's not a fully efficient process. We consume ATPs during this process, but it's much better than just letting that lactate go to waste. All right, so here's a summary of the Cori cycle. In general, right, we have glucose. It's in our muscles. It undergoes glycolysis. It generates pyruvate, and this can be during aerobic or anaerobic metabolism. During anaerobic metabolism, that pyruvate is converted to lactate. That lactate then again seeps out of the muscle tissue into the bloodstream and enters the liver, which is this lovely purple depiction here. The lactate's then converted back to pyruvate, back then through the process of gluconeogenesis, which we're about to look at, turned into glucose, and then recycled back into the process again. So in general, we consume a total of 6 high-energy molecules, 4 ATPs and 2 GTPs. They're consumed then during the conversion of pyruvate to glucose. So again, it does take some energy in order to make this happen. So the overall reaction to pyruvate plus 4 ATP plus 2 GTP and some of our reduced NADH even are then again making glucose, ADP, are then our lower energy molecules, 6 inorganic phosphate molecules, and our oxidized NAD. So what does that really mean for us? Well, it means that we're spending energy, but ultimately we are gaining a lot of net energy, because if we then take this glucose, that's the product here, we can generate a total of 30 ATPs, right? And so if we're spending 6 to get 30, that's still a big net swing. So that's why we ultimately have this process. All right. So let's look then at gluconeogenesis. How do we go more specifically from pyruvate or other biomolecules into glucose? So gluconeogenesis is the metabolic pathway. So it's a series of enzymes. Every time you see pathway, think series of enzymes by which glucose is synthesized, again, from these non-carbohydrate materials. This is an anabolic process. Remember, anabolic means we're building. Catabolic means we're breaking down for energy. So anabolic process consumes energy, and as we just saw in the last slide, we have 6 high-energy molecules that are consumed. And this only occurs under specific conditions. We wouldn't generate glucose from other biomolecules unless we really needed to, because this is a fairly high energy expenditure. So this occurs when, again, we have a lot of lactate building up in the blood because of exercise. This occurs then when we don't have much glycogen in our cells, and so we really need to generate more or maintain our blood glucose. So this occurs when you become exhausted from depleting your glycogen stores, and ultimately then this can happen to replenish the liver glycogen stores when it needs to. All right, so the potential starting reactants, so the starting materials are pyruvate, lactate. As we saw, lactate can be converted to pyruvate, glycerol, and certain amino acids. All right, so in any case, pyruvate is considered to be the initial substrate for the gluconeogenesis pathway, but we can feed these other molecules into pyruvate. We can convert them to pyruvate and then generate, again, glucose. All right. So it's in some ways the opposite of glycolysis, but there are some enzymatic steps that are different. Some enzymes are reversible. Some enzymes are not reversible, meaning some enzymes only produce the products, whereas some enzymes can generate reactants from products or products from reactants, so they're reversible. So in the case of gluconeogenesis, it's not exactly the reverse of glycolysis because there are 3 different total enzymes involved, but many of the same enzymes are involved, and many of them, and it does still occur in the cytosol of cells in the liver. So here's what we mean. Going down, this is the process we looked at on Wednesday of glycolysis, so taking glucose, generating pyruvate. In red here are the processes that are different for gluconeogenesis. So going up now is gluconeogenesis. So going from pyruvate to phosphoenol pyruvate, we have an intermediate then of oxaloacetate. In red here, then we have a different enzyme which requires ATP to do this conversion step. Step 6 through 8 are the same. There's nothing that's changed about them other than if we're going the other direction, we're consuming ATP rather than generating it. We have a different enzyme here. So this enzyme, phosphofructokinase, is not reversible. We need a different enzyme. So here we have, again, this new enzyme. Step 10 is the same as step 2. And then finally, the very last step, this hexokinase is also not reversible. And so we have then glucose 6-phosphatase generates glucose from our glucose 6-phosphate. So overall, if we have a step that consumes ATP during glycolysis, it produces ATP during gluconeogenesis, and the opposite is true as well. All right, so we might need to store glucose for when it's really needed, when we're running low on blood sugar, and we need to generate more sugar to keep our muscles moving. Well, we have, again, a polymer called glycogen. Glycogen is a branch polymer of glucose. So it differs in the other polymers of glucose we've been studying in a couple ways. Remember, we have cellulose and chitin, both of which are polymers of glucose, but those have different bond types. We have a beta-glycosidic link instead of an alpha-glycosidic link. Humans have problems breaking. The other difference is glycogen is also branched, so it goes off in a bunch of different directions rather than just being a linear molecule. So this is how we store carbohydrates in human and animal tissue. And this is primarily done in the muscles and in the liver. So it makes sense we have it in the muscles. We're going to be moving around. If we run out of sugar in our muscles, well, it's going to be an issue. Similarly, our liver helps regulate our glycolysis and gluconeogenesis, and so it's helpful to have stores there. So glycogen is very helpful for storing energy. How are we going to actually make it, though? We make glycogen from a process called glycogenesis. That is exactly what it sounds. We're generating glycogen, and we're synthesizing it specifically from glucose 6-phosphate, which is the very first intermediate in the glycolysis cycle, or the last step in gluconeogenesis. In any case, to make glycogen instead of glucose, what we're going to do is we're going to isomerize that glucose 6-phosphate, turn it into a different isomer. We're going to activate it by adding inorganic phosphorus, and then we're going to ultimately link it together. All right, so let's look at these steps. Step 1, isomerization. What we're doing is we're changing glucose 6-phosphate to glucose 1-phosphate so that we're able then to perform the next step, which is activation. So now we have glucose. We activate glucose 1-phosphate by the attachment of UTP, another high-energy molecule. This is uridine triphosphate. It's, again, analogous in many ways to ATP or GTP, but it has a much smaller role in the cells for transporting energy. So what we do is we ultimately form then a UDP, so a uridine diphosphate, glucose intermediate there. So here we have our glucose 1-phosphate. We have our GDP or UTP molecule, These two inorganic phosphorysis phosphates leave, so those leave the reaction site. Then our glucose gets attached here, and so we have now a UDP glucose intermediate. All right, and finally, step 3 is linkage to the chain, so how we're actually growing our glycogen polymer. Ultimately, then, that UDP is released and it's converted back to UTP to start the process again, converted back to this molecule, and that last step consumes 1 ATP, so it costs energy to build glycogen. All right. So this ultimately what we're doing, we're generating then to regenerate our UDP. These separate. We now have our glycogen polymer, which we're really after. We have our UDP. That UDP gets regenerated. All right. So the opposite can also happen. Glycogen can be degraded, which is extremely useful for needing energy. We've built up all this store of energy as glycogen. Well, now we're going to be able to use it. Ultimately, glucose 6-phosphate is produced from glycogen. And it's not exactly then the reverse of glycogenesis, but it's similar. It does not require the UTP, UDP intermediates. So instead then we have direct phosphorylation right away, which removes a glucose from the glycogen unit, and we make glucose 1-phosphate, and then it gets turned back into glucose 6-phosphate. All right. I don't think we'll look at that in much detail. It's good to know the overall process, though. Here we go. In glycogenesis, we have our glucose gets converted to glucose 6-phosphate, isomerized to glucose 1-phosphate. We have our UDP intermediate, and then finally we're at our glycogen stores. So we're ready to go whenever we need energy. When we need energy, the inorganic phosphorus attaches to glycogen. And so then we have now one less molecule on our glycogen chain. We've pulled off one glucose. That's then glucose 1-phosphate right here. So we skip this intermediate step going back the other way. Generate glucose 6-phosphate and enter glycolysis. So we have regulation of the carbohydrate metabolism, just as we have regulation of all metabolism by various mechanisms. So we're going to look at 3 main categories of carbohydrate regulation. First one is insulin. So insulin is a 51 amino acid protein hormone. It's a little protein, and it's excreted by the pancreas when there are high blood glucose levels, and that lowers blood glucose levels, ultimately by binding to receptor sites on the cell, which encourages the cell to uptake glucose and process it. We also have another regulation method called glucagon. It's another, in this case, it's considered a polypeptide rather than a protein, although it's just, again, a chain of amino acids. There are 29 of them. It's also produced in the pancreas, and it's the opposite of insulin. So it's released when blood glucose levels are very low. And so what that does is it increases blood glucose levels by ultimately speeding up glycogenolysis, right? Generating glucose from glycogen. And the last main regulator is epinephrine, which is more commonly known as adrenaline. It's released by the adrenal glands in response to a flight or fight response, right? A flight or fight situation, anger, fear, excitement, etc. And it really works in a similar way to glucagon, except it's much more rapidly. And its primary target is muscle cells, so you're quickly able to uptake a ton of glucose in order to then be able to expend that energy quite rapidly. All right, so we can regulate carbohydrate metabolism now. Just to quickly summarize, what do we really want to know in this chapter? Well, what we really want to know are the products of these various pathways, the reactants, and really what it takes to get us there. So we want to know glycolysis. We want to be able to realize what that's generating. We want to kind of know how it's regulated. We want to know how we build glycogen. We want to know how we regenerate glucose. We want to be able to recognize all those pathway words. So glycogenolysis, it's not a very good word. but gluconeogenesis, etc. We want to be able to recognize all of those, know what they do, and know what their consequences are for carbohydrate metabolism. All right, well, thanks for tuning in, and I'll see everybody on Monday.